Welcome back to General Ecology. Today's segment, and well, there's 4 segments coming in here on mutualisms. And as I said at the end of the last segment, this is a topic near and dear to my heart. It's an interesting topic because it has not been modeled very well, and so has not been really treated very effectively in ecology. But increasingly, we're becoming aware that the mutualistic interactions are very important in our systems. Before we get into the chapter, I'm going to have my first slide here is sort of the tail end of the competition presentations, where I went over all of these equations and how we developed them going from the geometric down to the competition. And in the competition equations, I had the wrong well, it wasn't wrong. I just didn't include the R1, N1. I had this segment, which is basically a development of this logistic equation, 1 minus n over k. Now it's 1 minus n over k, but you add the competition coefficient. That is the competition from species 2 on species 1. And same thing over here, R2, N2, 1 over, and here it's the competition of species 1 on species 2's carrying capacity. So just wanted to make sure you have that. Make sure you understand why we have fleshed this out and why this brings in competition, right? Otherwise, it's the same as the logistic, 1 over n over k. Now we've added competition. Okay? And of course, you have to have it for both species, species 1, species 2. All right. Excuse me, mutualisms, chapter 15. So one of the things that we have to think about when we talk about mutualisms is that it's basically species interaction, two things living together. I'm going to come back to this word symbiosis in a bit, but living together, sym-bio. Okay, actually that would be together living. And the initial coining of the term in the late 1800s was by a lichenologist who you'll see, where I'll talk about a little bit later, did not separate mutualism from parasitism within the word symbiosis. Today we tend to use it specifically, or tend to use it for mutualisms, almost analogous, or it's the same word, but no, it's really, I think that does a disfavor because it makes it seem like mutualisms are a thing and that organisms are basically seeking to make other organisms happy. And evolutionarily, that makes no sense. What an organism is doing is seeking to make itself happy and to perpetuate its kind. Okay, it's not saying, gosh, I really like those fungi on my roots. I think I'll support them because they're really fun. They're good things. No, it's in it for itself, and I'll explain that a little bit later. And the original concept didn't really get into mutualism and parasitism per se. It said, yes, they occur. Sometimes it's more mutualistic in a symbiosis. Sometimes it's more parasitic in a symbiosis. So anyway, it's a few of us in the world are really upset about this changing of the word to mean mutualism, and it's not helping. All right. Here is a nice diagram showing you have two different species, species 1, species 2. When they both are benefiting, getting a fitness enhancement through their interaction, that's a mutualism. If only one is benefiting, then it's a parasitism, and it can go from species 1 to 2 or 2 to 1. So in terms of mycorrhizal fungi, the fungus may actually parasitize the plant. Or I'll show you an example of plants that parasitize the fungi. All right? So you're constantly shifting back and forth along that arrow between mutualism and parasitism, and what's not included in that figure is the environmental construct. So what's going on in the environment may shift the interaction more towards parasitism by a species, or mutualism, or parasitism by the other species. It's just something to keep in mind and help clarify what these interactions are about. All right, so here is an example out of your book, jumping way ahead to figure 1717, and it shows this particular damselfish and cleaner fish on the damselfish. And so basically, if the cleaner fish are around and there's a lot of parasites on the damselfish, then they clean those parasites off. And their meals are primarily made of those ectoparasites. Okay? So when there's a lot of ectoparasites, that's what they're eating. When there are not many, they're not eating many ectoparasites because there aren't enough to go around. And now if you look at their stomach contents, you find that there's mucus and scales from the damselfish. So the cleaner fish start eating the damselfish skin, scales, mucus, because there isn't enough ectoparasites around, right? There's only 0 to 1 per fish, and therefore the cleaner fish start cleaning the fish themselves, not cleaning the fish off, getting rid of the ectoparasites. Hope that made sense. So cleaner fish eat the ectoparasites when abundant. They'll start eating damselfish, scales, and other things if the ectoparasites aren't. By the way, that picture is not a damselfish. That's a sunfish being cleaned by cleaner fish. If you've never seen a sunfish, you're missing out. They're bizarre. I'll just leave it at that. Okay, here is a story that I think is very interesting, and how we can apply sort of the mutualistic ideas to a conservation problem. So on these islands, I don't know how to pronounce that correctly, I'll be honest, Mauritius Islands, there is a tree that is going extinct. And someone figured out that on that island there used to be some birds called dodo birds. You know, this was a species that went extinct through human interventions, basically killing them off. Well, with the dodo birds went their consumption of a number of tree host seeds on these islands. So those dodo birds were removed, basically eaten by humans, and the seed were no longer being eaten by the dodo birds. The seeds were not passing through the digestive tract of the dodo birds and were not germinating. And so slowly but surely, those tree species were dying, the adults, and were not replenishing, regenerating. Some of these are really interesting trees, including one near and dear to me is the ebony, which yields some very hard wood, good for piano keys and guitar fretboards. So we need ebony. We need ebony. So what these researchers did is once they figured out that probably the lack of the dodo bird digestive tract was causing a reduction in germination, they said, well, maybe we could bring in other animals to see if they might help get these trees to germinate. And so they tried turkey, but that didn't work. And then they brought in these tortoises, which were also on the island, I believe, or at least nearby. And they do consume the seeds, and they pass the seeds intact. I mean, a low percentage are viable, but enough that you can actually get the population of trees back by introducing this tortoise back into the system. And now we have the seedlings starting to regenerate again, which is a great story. I mean, it's putting all the pieces together, coming up with a hypothesis and experimental design, trying it out and saying, yeah, that works. Then you've got to decide if you want to manipulate the system, because maybe that tortoise, maybe I'm wrong, maybe it did not belong on that island. But it's worth a shot now. When they did the experiment, according to your book, by the way, I believe it was they used a very small island that they could manage in case things started to go awry, but they didn't. Okay. Good story. Mutualisms. All right. They are important, as that last example explains, at least for the tortoise, for the tree, maybe for the humans if they play music. This concept has largely been left out of ecological discourse. I'm showing you here a bunch of slides. There's an orchid, the clownfish, there's the anticacia, there's the yucca moth symbiosis or mutualism, fig and fig wasp, lichens, leaf-eating ants, leaf-cutting ants. And if you talk to an ecologist who isn't really honing in on the ideas of mutualisms, They're going to say all of these are special cases. They don't really matter. I think lichens may be the one that they'd have to say, yeah, you're right. Actually, lichens are everywhere, aren't they? Maybe they do matter. Maybe they're the first line of a succession in a new habitat that's just been exposed through volcanic activity or something. But in general, these others are special cases. I mean, clownfish and the sea anemone, I mean, that's just special, right? Very unique situation. I'll talk about that in Greekum orchid on the left side in a bit. And the yucca yucca moth, I mean, they're just species to species interactions. They are mutualistic, but ecologically not so important to some. I'm going to argue that they're very important. So let's go down the path first to just look at more of these examples of, quote, special cases. Here's the ant acacia ant story. And you could see on the panel on the right, the more ants you have on the acacia, the less herbivorous insects. So if the ants are there, they eat the insects and the acacia survives better. The acacia produces these little houses. You can see this, and the ants go in and out of those little holes and then just kind of go up and down and kill off and eat the herbaceous insects so that the acacia survives better. Not only does the acacia make these interesting thorny houses, but also makes these little protein packets at the end of all their little leaves that feed the ant. So the ants are getting fed by the herbivores, insects. They're using those for nutrition, but they're also getting extra protein from the plant. So the plant is keeping them there in a sense. Does the plant know that the ants are getting that? I'd argue no. But evolutionarily, if a plant was producing these protein packs, it had a better chance to survive because the ants were getting rid of the herbivores. It didn't know, but it was happening. And so it continued to produce, each new generation produced more and more of those protein packets, right? Clearly, the presence of the ants increases survival also of the acacia, right? All right. Here's another one near and dear to my heart. I had a committee member on one of my dissertation, my PhD, who had a habit trail for hamsters in his office, and that habit trail housed leafcutter ants. And they had their little sections where there was the latrine. I think in here it's called the dump chambers, where they got rid of the waste. They had where the fungus was being maintained in an agricultural sort of setting. The fungus produces food packets for the ants. The ants are constantly chewing up the leaves and then feeding them to the fungus. The fungus lives on the leaves and produces little droplets of food for the ants to keep them there. So there's a whole storyline here. If you haven't looked into it, it's pretty amazing. In this guy's office, he left the window open, and the trees, certain trees around his office on the Berkeley campus were defoliated periodically because the ants would hone in on that tree and then take all the leaves because they were considered, somehow they figured out that their fungi really liked that particular species. Never found out if the grounds folks were happy with that or not. Right. An additional feature, by the way, is that the ants carry antibiotics, actinomycete bacteria on a little spot in their carapace on the underside that is keeping their fungal colonies clean. So they're actually using antibiotics to keep their fungal colonies pure and make sure nobody gets in there and starts to ruin their cultures. That's usually what you see when you're in the forest. You see a trail of ants carrying their little pieces of leaf. Sometimes you see other ants riding on those, so they're keeping other things away while the big guys are carrying them back to the nest over here on the lower left. Is this a mutualism? Has it evolved together, so to speak? Yeah, I think so. It's not co-evolution per se, because the ants aren't really, maybe their behavior is changing and maybe the fungus biology is changing, but it's just they're associated with each other. It's a hard thing to separate out of coevolution, but some would argue strongly it's not co-evolution. You don't see a fungal trait causing a different fungal trait in the ants and vice versa. So it's not co-evolution. But you have a fungal phylogeny here on the left side. Okay, so russulas, boletes, tricholomas, cortinarius, these names may ring true for some of you. And then the lepiotas, from there, you get the leafcutter ant fungi, the fungi that the leafcutter ants are colonizing or growing, I guess is the best way to put it. And over here you have the ant phylogeny. And so here you have all the ant names. In a couple of cases, it's the fungus name because there's no ant associate with that fungus. But otherwise, it's the ant name on this area. And then down here, it's the fungus name if there's no ant associated with it. And the cool thing, I think, to note is like up here in the G1 group, so note up here it says G1, you have the Atta ants, and look at the branching pattern. Pretty much, I mean, it's not a perfect congruence, but it's pretty close. The same thing happens down here in the G2 group. Nice congruence between the two branching patterns, fungus species and ant species. And then, but the middle one, G3, it's all over the place. Look at the ant interactions here. It's all over the place. There was maybe some nice evolutionary story on the fungi, but over here it's all mixed up. And there's a good story. Up here and down here, when a queen starts a new colony, she takes a little packet of the fungus with her and starts the new colony with that fungus. So there's always this sort of one-to-one relationship in ecological time between ant and fungus, and therefore they have evolved in a very similar branching fashion, if you will. These guys don't do that. They harvest and establish new colonies each time they start a new colony. The queen starts a new colony. So they're picking them out of the environment, and it's kind of, yeah, they're related species of fungi, but they're not the same clone as the G3 and the G1 group are doing. So that's why those top and bottom branching patterns are so similar. Okay, moving on. Darwin saw this orchid, an Angraecum sesquipedale, in the tropics. I think it was in maybe Madagascar. And it has a very long spur. You could grow these. I had one of these growing in my collection back in the day when I was in San Francisco, and the climate was good for orchids. Way down here is a little nectar pool. And so he saw this. He said, well, some moth, because it's white, that tends to be night-loving insects and birds and bats and things like that. Something has a really long tongue to stick its head in here, get the pollen in the process, but then get a nectar reward way down here. And he made that prediction. He said there's got to be something out there with a really long proboscis, and it was found 41 years later and actually caught in the act here in 1992. Now remember, Darwin was down there in 1860s, right? So he made the prediction, didn't get to see it come true, but there it is. I forget the name of the moth. I apologize to the entomologists. But this Angraecum definitely has a very good partner in their symbiosis. Pollination strategies. Now, pollination is a really big mutualism, as you're aware, and there's a lot of work being done. Most of the pollination interactions aren't like this Angraecum, where it's one-to-one. There's one moth on one orchid. Okay. Usually, it's a little bit more complicated, and sometimes really so. So these stars represent, let's see, the circles are animals that have lots of different mutualists that they're interacting with. Here is a plant that has lots of different mutualists that it interacts with. Others a little less so. There's only two here, okay? But you get the idea. These networks are complicated. They're hard to sort out. A bee will visit all sorts of different species if you watch it. And that makes for a really difficult thing to model, as mutualist modelers are discovering. But it's not unique in the mutualism world that, if an insect rejects a pollen reward, it maybe is less fit. So how specificity evolves where they stop taking pollen if it's available is an interesting field of science, evolutionary science. Here are a couple more examples of mutualists. That moray eel has a cleaner wrasse. We've talked about that in the last section. It's not going to get eaten, and that eel seems to be quite happy with that wrasse cleaning up the last meal, I suppose. This little dude is really cool. This is a little, oh my gosh, nudibranch, I believe, kind of a sea slug. And all those little green dots, I don't know if you can see them, it's in your book, are chlorophyll packets that it eats algae and then incorporates those algae into its little tendrils there. I don't know what the magenta tips are about, but this little nudibranch is actually photosynthesizing, not on its own, but with a mutualistic interaction with these chloroplasts, I suppose, that it picks up from algae. That sentence will make more sense in a little bit, where I said mutualistic interaction with the chloroplasts, not the algae, but it eats the algae to get the chloroplasts. So what's a mutualism there and what's a parasitism? All right, there are three kinds of mutualisms that we kind of pigeonholed all interactions into, and you can think of your particular species interactions and see if you can't figure out if it's trophic, defensive, or dispersive. So trophic is whenever you're getting energy or something out of that interaction. Nitrogen fixed from the atmosphere for plants. We breathe in this atmosphere, it's nitrogen, we breathe it right back out. 70% N2 gas right back out. But a bacterium, a prokaryote, can take that nitrogen and make it into ammonium. And now we can go down the path of making amino acids and things like that. Our gut bacteria is trophic, obviously. They're breaking down stuff and releasing energy that we then absorb through our intestinal lines. They get some of it, though, so they're benefiting. Bees and flowers and other pollinators, including bats, not so much rats, and other insects and moths and things like that. Mammals and fruit and seed dispersal. Sorry, they're eating them. It's not dispersal, it's trophic. I put there corn and humans because, you know, corn doesn't exist on its own. We have fields and fields and fields of corn. Corn has figured out how to make us want it. And so we make a lot of corn. And is that a mutualism? Do we get something from the corn? We do. So depending on, you know, if you're like an alien landing on this planet, you're going to go down to Iowa and you're going to go, whoa, look at all this corn. And then you say, well, gee, can't the corn live on its own? No. If you put a corn seed out in your backyard or out in the forest, it'll germinate and die. No matter how much light you give it, it's not going to do anything because it can't survive on its own. We've basically tweaked it to this thing that we eat in our picnics and things. Okay, defensive. We've already talked about the Anticasia, the wrasse fish and other fish, Nemo, our clownfish and the sea anemone, and dispersive, plant, pollen, and also seed dispersal with mammals. Some tight mutualisms with those different types. Sometimes it's intermixed. There's both a trophic, where the seed provides some food, but then the animal is dispersing other seeds for that plant. So you've got both functioning, for instance. Now, I really want to make this point, and you're going to see it in the next segment. Mutualisms are not special cases. Mutualisms are ubiquitous. So in the next section, I'm going to dig into this a little bit and hopefully convince you of that fact. All the stuff I've been talking has been kind of the, wow, that's beautiful. Look at that nudibranch with all its chlorophyll, chloroplasts, and all that. Absolutely gorgeous. Oh, it's a special case. All right. Maybe. Look at lichens everywhere, right? It's evolved a number of different times. This is not reindeer moss, which is a reindeer moss is actually a moss. This is a lichen, Usnea. Grows on the oaks in my old stomping grounds in California. British soldiers they're fruiting here. Those little red things are the ascomycete fruiting. It's like a cup fungus. If you look at it carefully, these are cups. And then, of course, there's crustose and foliose lichens, etc. And you may not know it, but there's also basidiomycete lichens. So these are guild fungi on the far right. Most lichens are made with ascomycetes, but you can also get them with basidiomycetes. There's a couple. They're actually, a lot of them are out there, but they're not as ubiquitous as the ascomycete versions. They are associated, the fungi are associated with bacteria that can fix nitrogen and carbon, so they're both photosynthesizing and nitrogen fixing, and therefore it's a three-way interaction and kind of a nice setup if you can get it. Okay. Mitochondria. I don't know if you're aware of this, but they're sometimes called endosymbionts. Every eukaryotic cell, almost, you never say ever, everyone, but every eukaryotic cell has mitochondria. If you look at the mitochondria with genetic tools, you discover it's got DNA that's in a looped structure. That's a prokaryote. That's what prokaryotes do. Okay? And then if you look further and watch those mitochondria divide, they divide through cell division just like a bacterium does. So these mitochondria are bacteria. They were basically sometime many, many years ago, there was just prokaryotes and archaea bacteria and prokaryotes, and they became eukaryotes with a symbiotic interaction. Are these important? Yes. That's how we get our energy, right? Every eukaryotic cell has to have bacteria in there making its energy. What do they get out of it? Maybe a house, maybe a place to live. But we're also feeding them. We're delivering ATP. We're delivering sugar. They're delivering ATP back to us. So they get something out of it. What else? How about chloroplasts? Also bacterial symbionts. Here you've got actually the genome of a rice chloroplast DNA showing you all the different markers that they have found on that rice genome. That's inside those chloroplasts. It is bacterial. Loop structure, cell division, same stuff. Here's another one that's sort of, if you were not careful with your evolutionary thinking, you'd say this is on its way to becoming an endosymbiont. So the rhizobia in the legumes are fixing nitrogen, all right? It's a bacterium. And it's kind of interesting because the plant makes these little houses, and the bacterium can actually live free living in the soil, but it doesn't do as well. When it gets into these nodules, then it takes off and does really well. Now these nodules are all red compared to the plant tissue. And if you pick up a clover or something in the legume family, pea plant, do it enough, you'll see the nodules. And if you cut them open with your fingernail or a knife or something, you'll see that they have a little red section, if not completely red the way these are. Why? And this is where it gets really interesting in terms of evolution of the symbiosis. So the bacteria are fixing the nitrogen. They're taking gaseous nitrogen and making ammonium. Huge. Only bacteria can do that. We say it's a nitrogen-fixing plant, but the plant isn't doing it. The bacteria that it has in its roots is doing it. Okay? That's an important factor. But the bacteria need it to be relatively anaerobic. They can't do it if there's a lot of oxygen around. So the plant in its nodules is creating a low oxygen environment in the nodules, and that supports the nitrogen fixation. So that's kind of cool, but I think the real story here that I want you to get is that inside there's something called leg hemoglobin. It's for legume hemoglobin. So our bodies have it too. That's why our blood's red. It's a multi-structured protein, 4 parts. It has iron. It looks red, especially if it's bound with oxygen, right? And then this is a very similar structure, only it's produced by legumes. The cool thing is part of the DNA code is in the rhizobium DNA, and part of that DNA code is in the legume DNA. And so here we have this sort of beginning of communication and the development of a very tight symbiosis the way mitochondria and chloroplasts are, because they too have genes that are both in the eukaryotic genome and the prokaryotic genome that make them work. And here it's developing in the rhizobium situation. And I think that's fascinating. I don't know if this is a sign that this is heading somewhere. It's successful, certainly. Are we going to have endosymbiotic nitrogen fixing down the road that is tightly linked and you really can't live without your host? That is, if you're the bacterium. Who knows? We won't be here long enough to know. And evolution doesn't quite work that way, just to be clear. But this is kind of an interesting sort of phase or point where it's not quite to the level of the mitochondria, the chloroplast, but it certainly is looking interesting, the nitrogen fixing. There are over 200 plant species and 8 families that support nitrogen fixation in this way. Of course, the pea plants and the Fabaceae with rhizobium. Betulaceae, alder, are associated with Frankia, a different bacterium, but also fixes nitrogen. And you probably know that alder is a nitrogen-fixing species, but it's not in the pea group. Rhamnaceae, Mycianothus out west, associated with Frankia. I'd find these massive nodules that would basically the size of the palm of my hand, and I would, what the heck is this? I thought they were mycorrhizal at first, but then I did some work and found out that they were Frankia nodules. Casuarina, that's this slide. I've already shown you this. And Maricaceae. So all of these are with Frankia, named for this guy named Frank. We'll come back to him in a minute. But a lot of species. And it turns out that it has evolved because nitrogen's such a limited nutrient out there. The plants have become become associated, mutualistic, with things that can make nitrogen for them. Okay. And here we go to Frank. I will take a pause here, if I may. Let's see. Let me go back to this. I'm confused where I am here. I thought there was a gap somewhere along the line here. So I'm trying to reconnoiter. Yeah, I should probably, let's see where, oh. Yeah, that's why I was, oh, there, I didn't see that gap up at slide 14. Damn it. All right. Okay, I'll go back to this slide and just finish it. I know where I am, yeah. So sorry. Okay, so now we get to this guy Frank. So we just learned about one genus of nitrogen-fixing bacteria that are on a lot of different plants called Frankia, so named for this guy. So you can see right in the name of this photo, who took the picture, it was Frank, 1894, all right? And what he discovered, not discovered, but he said, hey, there was a guy named Hooke who had come up with this new contraption called a microscope. And then there was a guy named Hartig later who wrote the textbook on plant pathology. And he said, there are fungi down in the roots of these plants and they're all pathological. They're all pathogens. We find them with our microscopes, they're all bad. Standard sort of scientific approach. You see it, you call it bad, and then you can't believe it's mutualistic. They didn't even know the word yet back then. So Frank, though, has all these pots with soil that he collected from the field. These pine seed were put into unsterilized soil. Okay, they grew pretty big. Okay, next to them, same soil, but using Pasteur's methods, steam sterilized, and those plants don't grow as big. And he showed that if you look under the microscope at the roots, there are no fungi in the roots of these sterilized ones, but they were loaded over here. In fact, you don't even hardly need a very powerful microscope to see all the mycorrhizae, that's where we're headed, that are on these pine seedlings. These were spontaneously, probably some spores landed in his experiment. So he had a couple, he looked in there, and indeed they were mycorrhizal, but not as intensely mycorrhizal as these on the far right. This is back in 1894, people. We've known about this symbiosis for many years. A.B. Frank saw it in the 1890s, and he called out Hartig and said, no, nope, nope, nope. Those are not pathogens. You don't see any rejection of the fungus by the plant cells. They are accepting them. Okay? So it's either an incredibly crafty pathogen or the plant wants that fungus there, or at least is benefiting from the fungus. And Frank comes up with two new words, mycorrhiza. So this means Myces is an island and culture in ancient Greece that had a mushroom cult. Still occurs today in San Francisco. No, so the mycorrhiza, so this is fungus, and then rhiza is root. All right, two sort of Greek words smashed together into a pseudo-Latin word. It's really not proper, but it's what we have, so it goes. Fungus root. He also comes up with this word called symbiosis. And at the time, there were a number of people that were starting to see this idea of things living together. And there was all sorts of strange languages, not strange, but there was Germans, as was Frank. There were Italians, and they were starting to put those two roots together, sim and bio, living together. But eventually we come up with this word symbiosis. And one of my graduate students this past year dug up the original paper by Frank. Everybody cites it. She dug it up, and my wife, who's German, and myself, and Claudia Viktoroff, the three of us have been working on making a direct translation of this old German prose, showing that he was probably the first to use the term and the concept of symbiosis, although it wasn't Englished yet as that spelling occurs. But de Bary was the other one that people often cite. Frank, by the way, studied lichens. So it wasn't a big shift for him to say, these plants and their fungi they seem to be living together. So, before we get to the next segment where I'm going to dive in heavily, I'm just going to give you a couple of terminology words that I want you to have in your back pocket when I go down to the next segment. Mycorrhiza, I've already defined as fungus root. Hypha, single thread of fungus. In plural, you would say hyphae, A-E at the end there. Again, Latinizing it. And then the other way to say lots of hyphae is to say that's mycelium, a collection of hyphae. And it can be very well defined as a mushroom or a truffle, or kind of loosely defined as a mat of stuff like cotton, or down into rhizomorphs, which are cords that allow for mass transport of flow and flow of water with nutrients between various parts of the fungus body, but also from root to root, and from the plant with its sugar back out to the fungus body. We find rhizomorphs quite often. Why do we think this is so important once we start looking? Here is a radish seed. Radishes don't form mycorrhizae, and I won't get into that. Take my class if you're interested. But those are really well-defined root hairs because they don't form mycorrhizae. They need to explore the soil better. But all around in this region, if it was just looking right in here, this radish root would very quickly deplete any water or nitrogen from the soil right in here because it would take it up, and it would be this sort of vacuum of no nitrogen, no phosphorus, no water. It's all been taken up by the plant, but it only goes out a couple, like a millimeter, if that. So the plant begins to evolve, or does grow, these root hairs, which get it out to a few millimeters. And now the depletion zone is a little bit broader. Okay, and that's obviously a benefit for that radish seedling. until you realize that most plants don't produce root hairs, especially in the ectomycorrhizal world. It's a little more common to see them in the arbuscular mycorrhizal world, and these words will make sense in a minute. But look at this pine seedling, the main one here. Here are its roots. One branch there, one branch here, long branch down here. All the little nodules are mycorrhizal root tips. And the hyphae are getting all that sugar from the plant and then going out and exploring the soil and getting nutrients, water, and everything, and bringing it back to the plant. Plant is photosynthesizing, bringing it back out to the fungi, or delivering it to the root tips, which then deliver it out for their nutrient acquisition. So in comparison, that little radish would take about that much soil. And that's why you don't see radishes and broccoli and things like that, those weeds, out in the forest, because they can't compete against the mycelial networks that mycorrhizal fungi have, and so they are quickly outcompeted and die off. So that brings us to the end of this segment, and next segment I'm going to focus specifically on mycorrhizae in actually two different segments to talk about their importance and you know how to see them and what they mean for agriculture, your backyard plants, things like that. So, cheers. We'll see you next time.